Hi guys, how are you? I promise you that today's class is going to be really quick. Okay, so this is the shortest chapter that we're gonna have in the whole huge book that we're studying. Chapter 10, Classification of Microbes. So, learning objectives for us here. Get my pen. Um, we have definition and difference between taxonomy and phylogeny, which are the three domains and the five kingdoms, which are the similarities between the groups that are in each one of the three domains, features of the organisms that belong to these three domains, eubacteria, archaea, and eukarya, which are the four kingdoms, and definition of taxa. So if we look at first, what's the difference between taxonomy and phylogeny? We have taxonomy as classifying organisms according to their similarities, and phylogeny is study of evolutionary history of these organisms. So for example, you can say and group animals, for example, sharks and whales into, it's a group of animals that have fins and live in the water. And then you say, oh, so what do they have in similar? They have fins, right? Okay. So do you think phylogenetically they are super closely related? I hope you said no, because sharks have fins, but they're fish. They're not mammals. Whales are mammals, right? So you see how that would be just a way to use the taxonomy to, for example, divide them into a group in a clade. You can create different clades according to whatever similarities you're trying to use, but that doesn't make them phylogenetically close. Okay? So when I talk here about phylogeny, I'm going to point out, and when I talk about taxonomy, I'm going to point out. Okay? So we start with Linnaeus in 1739. Actually, yeah, that was the beginning of it when he divided all the organisms into only two kingdoms, which were Plantae and Animalia. And then later, other scientists found out that bacteria and fungi should not be put in the Plantae, and then they created another kingdom that is called Protista that we're going to talk about. And then in 37, the prokaryotes were introduced to distinguish cells that did not have a nucleus. So they were different from the eukaryotes. In 68, the kingdom prokaryote was characterized, and 69, Whitaker defined the five. That's my pen. I'm not even touching, see? And it's still writing. How crazy is that? The five kingdom system. Okay. Wois in 1978 had a very interesting observation that followed us until the recent days, which is when he was looking at ribosomal RNA sequences, he found that, what is that? Is remember that ribosomes have RNA. And that RNA that is present in the ribosome is super conserved. Meaning what? If you compare different bacteria, rRNA sequences, they're practically identical. So you don't have different species changing to match the sequence of ribosomal RNA. So this was a good tool to group organisms. And based on our ribosomal RNA similarities, they created three domains: the eukarya, the bacteria, or eubacteria would be the same thing, and the archaea. So if you think, wow, the archaea is such a small group compared to the bacteria and the eukarya, how can they have their own domain? Because all the organisms in this group have identical or very similar ribosomal RNA sequences. Okay, this is the point. Is this point important? Super important. Okay, so eukaryotes came from enfoldings of prokaryotic plasma membrane. And so why enfolding? Because remember how eukaryotes, all their organelles of cells that have a nuclei, the organelle is wrapped around pieces of plasma membrane. So this is what this is referring to. And bacteria developed into organelles in the eukaryotes. So when you look at this three-domain system that is shown here, you see how all this is the bacteria, right? What is that? Mitochondria. What is mitochondria doing here? Is mitochondria a bacterium species? Yes or no? No, it's not. So what is mitochondria doing here? And then there is a red arrow that comes all the way here to this point. What is that telling you? It is telling you, let me show you this slide here now. It is telling you this. It's telling you that an early cell one day incorporated a bacterium, and that bacterium became the mitochondrion of that eukaryotic cell. So it was kind of like an immature cell that once it incorporated this crazy bacteria that wanted to live inside it, that became the energy powerhouse of that cell. The same thing for chloroplasts. They were on one day a bacterium. This is a model, right? Nobody can ever prove this because it happened millions of years ago. But it's a theory that these two organelles were one day bacteria. Okay, what else do we need to see from here? See, and chloroplasts are here, and the arrow brings you to here. Okay. We also have, so this is one domain, bacterium. This is the other domain, archaea. Remember that archaea are extreme halophiles or methanogens or hyperthermophiles. Bacteria that need a high concentration of salt here, or that can produce methane, or that can live in super high temperatures like volcano temperatures. And then eukarya is the third domain, and inside this domain is where you have most of the kingdoms. So which are the kingdoms here? One kingdom is the bacteria plus the archaea, so it's the prokaryotes. So prokaryotes are bacteria and archaea, right? You see, I'm not touching and my pen keeps moving. And then, so one kingdom here. Which are the other kingdoms? Animalia, fungi, plants, and what are all these other organisms? Anything that I did not put a little red dot are classified in the kingdom Protista. What is the kingdom Protista? It's a catch-all group of anything that is an eukaryote and it's not a fungi, an animal, or a plant. Is it that simple? So even if they're not really close together or related, you still call them in the same kingdom? Yes. So that's the Protista, right? And now we're going to talk about differences between bacteria, the three domain, which are bacteria, archaea, and eukarya. So what I want to highlight, and this table is from the book, right? What I want to highlight to you. Archaea and bacteria, prokaryotic. Eukarya, eukaryotic, right? Obviously. Cell wall, peptidoglycan in bacteria, pseudomurine, you find that word in the book. You know my pen is not going to help you here to write. Pseudomurine is the name of the sugar that makes the peptidoglycan, that makes the cell wall in archaea, peptidoglycan in bacteria. And there are different types of carbohydrates and proteins connected that make cell wall in eukarya. Eukarya have cell wall? What kind of organisms have cell wall in the eukarya? Fungi, they have a chitin. Chitin, you're right. Right. Chitin is a protein. Chitin cell wall. That's for fungi. For plenty, you have for plants in the plenty kingdom, the cell wall is made of cellulose, right? So other than plants, and then algae can have cell wall too. Okay. So cell wall in eukaryotes. And now we're going to talk about, let's see what I want to highlight here. Well, the only ones sensitive to antibiotic are bacteria. Let's go to the other part of the table. I have here histones. You don't need to really pay attention to this, but it's proteins that the DNA coils around. So you find it in eukaryotes, but not in prokaryotes. But in archaea, yes, but not in bacteria. And this is important. The prokaryotic ribosome is 70S, and the eukaryotic ribosome is 80S. What does that mean? Interestingly, mitochondria and chloroplasts, remember they were one-day bacteria? So they have their own ribosomes with 70S types of ribosomes. So what's the difference between these two? The prokaryotic ribosome they do a centrifugation and they spin it down, and that sedimentation of material when they're isolating ribosomes gives this number. So just so you know where this 70 comes from, okay? So 70S is the combination of the 50S subunit and the 30S. Why am I telling you this? Because the eukaryotic ribosome is 80S. So what would be this useful for? Antibiotics can target 70S ribosomes and stop what? What ribosomes do? Protein synthesis. So if you target a prokaryotic ribosome with a drug, you stop this bacteria to grow inside your body. And at the same time, you're not affecting your 80S ribosome. So you're not stopping the host cell synthesis, only the bacterial cell synthesis. So it's a very selective mechanisms that help you get rid of microbes through antimicrobial. Okay? And remember, eukaryotic cells grow by mitosis and prokaryotic through binary fission. So what else I want to tell you here? Just want to see if I did not miss anything earlier. I don't think I did. Scientific nomenclature. So what is the binomial nomenclature? We talked about this before, right? The genus is always capital letter, Staphylococcus, for example, and the specific epithet would be epidermidis, lowercase letter, okay? Remember that the word needs to be written in italic or underlined. Okay, so if you're handwriting a name, let's say staff. Let me try again. Staff.
Just a sec.
My Siri is talking to me. Staphylococcus. So if I'm handwriting this, epidermidis, I'm just gonna abbreviate it. Epidermidis, I would have to underline it to make use of the binomial nomenclature, okay? So it's always underlined if you're handwriting. If you are typing, you usually put in italics, okay? First letter capital, second letter lowercase letter, okay? So letter that the specific epithet starts with, it's lowercase. So all this was phylogeny, right? Did I mention the kingdoms? I think I forgot. No, I did, right? So let's say again. So we have 3 domains, which are the bacteria, archaea, and eukarya. And inside the 3 domains, we have four kingdoms: prokaryotes, animals or animalia, fungi, plants, and protista. So these five are kingdoms. And all this, the three-domain system, is a phylogeny type of classification. Okay, so this is a phylogenetic tree. When we talk about what I'm going to show you now, which is this is a taxonomy study. So what is this? All the organisms are included in this big square right here, okay? And then you have the domain level. The domain level is the eukaryote. So all this is taxonomy, right? We're looking at RNA sequence, and we're looking at evolutionary aspects of these animals, of these organisms, including animals. So then the domains are here: eukarya, archaea, and bacteria. The kingdoms, so now I am just going deeper into this classification, for example, for fungi. So if I think about the fungi kingdom, I'm going to design a phylum. So there are different phyla inside the fungi kingdom, but one phylum would be the Ascomycota. And then here I have the name of the class, the name of the order, the family, the genus, and the species. What I'm trying to tell you is that the phylum is composed of several similarly in structure classes. The order is composed of composed of a small group of similar families. The genus is composed of a small group of species. So inside the Saccharomyces genus, you have different species, not only Saccharomyces cerevisiae. Okay? So each one of these groups contain smaller groups inside it. And here I'm just showing you one slice of this classification. So I selected fungi, and from all the phyla inside the fungi kingdom, I selected Ascomycota. Inside this big phylum, Ascomycota, I only selected class, Hemiascomycetes. So I'm just showing you how you narrow this down. So can I say that a species lies under the genus, and the genus is a classification that lies under family? So genus is always going to be smaller groups than a family group. A family is always going to be small, different families are always going to be smaller than one order, because the order includes all the families. Did you get it? Just want you to notice one more thing here. Now I'm doing a different type of taxonomic study here for archaea. But I want to mention that archaea and bacteria do not have a kingdom. They belong to the prokaryotes kingdom, but that's it. You don't have one specific kingdom for them, okay? Just for archaea. And then phylum. You have eukaryote, and then it keeps going down because you're talking about a methanogenic archaea here, and here it keeps narrowing down until you get to E. coli. So it's the same type of mechanism that I explained for you with fungi. So this is a taxonomic hierarchy. Remember that when you talk about prokaryotic species, they are a population of cells with similar characteristics. When you talk about eukaryotic species, they are a population of organisms, not cells, with similar characteristics. Because remember, all the prokaryotes are unicellular, right? Unicellular. A clone would be a population of cells derived from one single parent cell, and a strain would be a subgroup. So if we go here, after species, this is not really in the taxonomy hierarchy, but I would put that species can be divided into different strains. Is this important? This is super important, because this explains in part why, for example, you have E. coli that live in your intestines and they do not cause any disease, but then you have a contamination by E. coli, an infection or a food, you ingest E. coli from some food and you get sick because there are different strains. One can be non-pathogenic and the other one can be pathogenic. So this is just what I told you, what the Protista is, which is a catch-all kingdom for a variety of organisms that include fungi, that include anything that is eukaryote and is not classified in the fungi, animalia, or plantae kingdoms. Okay, there is a video here that explains to you, and also the grouping of Protista in this one kingdom is based on similarities on ribosomal RNA. Last but not least, you have viruses not being part of any domain because they don't have cells. Okay? So viral species have families, they have order, they have families, they have a class, but not sure if they have order. I think it's just family, class, genus, and species. Okay? Actually, not even species. They have usually just the genus name. Okay? So we're going to see that later. So they don't have any domain and they don't belong to any kingdom. Okay? They do not have a cell. So it's really hard to include, that's why they don't include viruses into this taxonomic and phylogenetic classification. Because they don't have a cell. They cannot even reproduce unless they're inside a host cell. Okay? And with this, we're done with this chapter. Super quick. Okay? Know this classification. Look at those learning objectives and you're good to go. That's all you need to know from this chapter.